Have you ever wondered what makes you, you? What is the secret code that defines every living organism on this planet? The answer to this riddle of existence lies in a molecule known as deoxyribonucleic acid, or more commonly, DNA. This intricate structure, coiled within the nucleus of our cells, is the genetic blueprint that determines everything from the color of your eyes to your susceptibility to certain diseases. Before its structure was discovered, DNA was a mystery, an enigma buried deep within the cells of all living things. It held the key to life, but no one knew how to unlock it. This molecule, we now know, plays a crucial role in genetic inheritance. It's the master weaver, intricately threading the tapestry of life with its coded instructions. The discovery of DNA's structure was a journey filled with twists, turns, and brilliant minds. But that, dear listeners, is a story for another scene. The path to DNA's discovery was paved with groundbreaking research and significant scientific breakthroughs. The journey to the heart of life's code, DNA, is a tale of curiosity, rigor, and sheer brilliance. This journey began in the mid-19th century with a humble monk named Gregor Mendel. In a small garden, Mendel carried out meticulous experiments with peas, breeding them for specific characteristics. Through his experiments, Mendel discovered that certain traits followed patterns of inheritance. His work suggested that there was a fundamental unit of inheritance, an unseen force driving the characteristics we see in living things. This was the first hint of what we now know as genes. Fast forward to the early 20th century, and we find ourselves in the laboratory of Frederick Griffith. Griffith was working with different strains of pneumonia bacteria, trying to understand what made some strains lethal while others were harmless. Through a series of brilliant experiments, he discovered a phenomenon he called transformation. Griffith showed that a harmless strain of bacteria could become lethal if mixed with heat-killed lethal strain. This indicated that some transforming principle, some substance, had been transferred from the lethal to the harmless bacteria, changing its nature. This transforming principle was a puzzle for scientists. What could it be? Many thought it was a protein, the workhorse of the cell, but a few suspected it might be something else, something more fundamental. The stage was set for a race to identify this elusive transforming principle. This race would lead to the discovery of DNA as the carrier of genetic information, a discovery that would revolutionize biology and medicine. These early discoveries set the stage for one of the most significant scientific discoveries of the 20th century. As we dive deeper into the story, we'll see how the pieces of the puzzle came together, culminating in the discovery of the structure of DNA by James Watson and Francis Crick. But that's a story for another scene. Stay tuned as the journey to the heart of life's code continues. In the early 1950s, the race was on to uncover the structure of DNA. This was no ordinary race, but a marathon of the mind, a contest of intellect and innovation. The finish line? The secret blueprint of life itself. In this thrilling scientific saga, an array of brilliant minds were vying for the prize. Among them was Rosalind Franklin, a fiercely dedicated researcher with a knack for X-ray crystallography. Franklin's work was pivotal, her X-ray images offering tantalizing glimpses into the unseen realm of the molecular world. Her photographs hinted at a helical structure, but the precise details remained elusive. Then we had Maurice Wilkins, a man whose contributions often go unacknowledged. Wilkins was a steadfast scientist, diligently chipping away at the enigma of DNA's structure. He was a key player in the race, providing crucial data that would eventually lead to the solution. But let's not forget Linus Pauling, an already established giant in the field of molecular biology. Pauling proposed a model of DNA, a triple helix that captivated the scientific community. Yet despite his reputation, Pauling's model was incorrect. His confident assertion, however, accelerated the race, pushing others to prove or disprove his theory. This was a time of intense scientific rivalry, a period punctuated by flashes of insight and frustrating dead ends. Each scientist was armed with their own unique set of tools, their own perspectives, and their own burning desire to be the first to unlock the secret of DNA. Yet, as we now know, the race was not to be won by a lone runner.
Instead, it was a team of two, a duo who would soon burst onto the scene, who would ultimately cross the finish line. Their names? James Watson and Francis Crick. These ambitious scientists were about to take center stage, their entry marking the beginning of the end of this thrilling race. This race was about to reach its climax with the entry of two ambitious scientists, James Watson and Francis Crick.
In 1953, Watson and Crick made a discovery that would change the world of biology forever. A discovery that would illuminate our understanding of life itself. They didn't work in isolation, they built upon the research of others, piecing together the puzzle that was DNA's structure. A significant piece of this puzzle was Rosalind Franklin's X-ray diffraction images. These images, dubbed as Photo 51, revealed a distinct, cross-shaped pattern, indicative of a helical structure. However, deciphering the precise nature of this helix was no simple task. Watson and Crick diligently pursued this mystery, sifting through mountains of data and wrestling with countless models. They were not just scientists, they were detectives, hunting for clues in the vast landscape of biological research. The turning point came when Watson had an insight about the arrangement of nucleotide pairs. He realized that adenine always paired with thymine and guanine with cytosine. This was the key that unlocked the door to the double helix. With this revelation, they were able to construct a model of DNA's structure, a double helix, two strands wound around each other like a twisted ladder. The rungs of the ladder were the pairs of nucleotides, held together by hydrogen bonds, while the sides were made up of sugar and phosphate groups. This was a structure that was both elegant and practical, allowing DNA to be easily copied for cell division. It was a momentous eureka moment when Watson and Crick finally pieced together the intricate puzzle of life's most fundamental molecule. Their model elegantly explained how genetic information could be stored, copied, and transmitted from generation to generation. Their discovery was more than just a triumph of scientific investigation. It was a revelation that would forever change our understanding of life itself. This discovery opened a new era in the understanding of life and heredity. It was a beacon that illuminated a path for countless future breakthroughs in genetics, medicine, and biotechnology. The discovery of the double helix has had far-reaching implications. Its influence extends across a myriad of fields, shaping the course of scientific inquiry and discovery. In the realm of genetics, the double helix opened the door to understanding how traits are passed from generation to generation, unlocking the secrets of heredity. Medicine, too, has been revolutionized. Knowledge of DNA's structure has facilitated breakthroughs in genetic testing, disease diagnosis, and the development of targeted therapies. Biotechnology has similarly been transformed, with DNA manipulation enabling everything from genetically modified crops to the development of life-saving drugs. Yet, the discovery of the double helix has not been without controversy. Questions about credit for the discovery have sparked debates about the nature of scientific collaboration and the recognition of contributions. The double helix, a simple structure with profound implications, truly is the code of life.